Hey everyone, today in this video I'll be talking about the non-symbiotic nitrogen fixation. Means it is also occurred with the help of some bacteria, okay, but there is no formation of root nodules in the leguminous plants. Okay, there is no formation of the root nodules on the leguminous plants. Rather, these type of non-symbiotic bacteria, they are present in the soil, and without the formation of these or without the need of these root nodules, they can fix atmospheric nitrogen, though in a very little amount rather than the symbiotic ones, into the soil. Okay, so this is basically non-symbiotic because there is no symbiosis or the mutual relationship between the plant and bacteria. Still, these type of non-symbiotic bacteria, they can fix atmospheric nitrogen, though in a small amount, but they fix, okay, just like the symbiotic ones. So there are two most important genus which are the true examples of non-symbiotic nitrogen fixing bacteria. One that is very important, that is azotobacter. Azotobacter azlei and crococcum. These are the two species predominantly, and another one is Clostridium. So both of them are motile bacteria, and these two are predominant genus present in the soil which can fix atmospheric nitrogen in a very little amount, but in a non-symbiotic. Remember this thing, non-symbiotic means they will not depending upon the plants for their nutrients. Rather, they only need carbohydrate in a huge amount as the source of their energy, okay, into the soil. It may be glucose, okay. Whenever glucose is present in a high amount in a media or in a soil, always remember this, that Clostridium will be produced at a very high amount. Whenever in place of glucose, if there is mannitol is present in the soil or in a culture media, azotobacter will be produced at a very high amount, or they will be predominating one. Okay, if you want to isolate Clostridium and azotobacter both, so you need to prepare a media, okay, that if it is supplemented with glucose, it will obviously give rise to Clostridium in a very huge amount. The count of the Clostridium CFU will be greater than the azotobacter, and if you replace the glucose with the mannitol in the same growth medium, it will give rise to huge amount of azotobacter. So this is the distinguishing feature. So this is a non-symbiotic nitrogen